Okay, so let's continue a series of lectures with Professor Pinna. Continue the lecture from yesterday, where he's going to elaborate why we are all fish, which will open the appetite. After the asteroid lecture showing all these ingredients in the asteroid, you feel like eating an asteroid. So,
thank you. Let me turn on this thing here. Okay?
Yeah.
Better? Yeah. My voice is not very good today. Well, you see Jack's not even here. Yes, this party was a bit too much in the drinking, so we'll survive. So it's good to have this thing here today. So don't overstrain my throat. Yesterday, we got a little carried away in the tree business, so we left a few things for today. So that's why I'm continuing the subject, because we just glanced over a few of the problems that need some more attention. And so I'm going to show some of the slides again. Actually, this slide is here because some of my fishy friends were curious about these numbers, and they said, oh yeah, we're going to discover the formula for that. And I said, okay, so I'm not going to tell you. But they're not here, so I guess they went back to the hospital. Yeah. I will show that to them later. But in case you guys are curious, let me turn on this slide here so I can write. More precisely so that I can write and you see. For those who were not here yesterday or who don't remember, this is the number of trees that you have according to the number of terminals or the number of species at the tips. Okay, so this number grows very fast. And as I said yesterday, when you reach 100, you have a number on the over there which is so big that it's bigger than the summation of all the particles in the known universe. So these numbers are super astronomical, as they call it in mathematics. And when we are trying to resolve phylogenetic relationships, what does that mean? That means building a tree for a given set of organisms. That's what we call resolving a phylogeny. Theoretically, you'd have to examine each one of them for the data and see how the data fits, how the data fits in each one of them, and choose the best one, which is sometimes not a single one, but several. There's maybe several optimal solutions. So obviously, there are no exact solutions for this problem. It is not a problem that can be solved algebraically. You have to solve it heuristically. And there are many methods. There are people who dedicate their entire careers to making programs about phylogenetic analysis that allows us to build trees, even in very complicated situations involving hundreds and hundreds, and even thousands of terminals of species. And this is a field that is, of course, very narrowly specialized. But the programs are available, and everybody uses them, and apparently they work fine. At least they work fine as far as we can test them, they work fine. Yeah. Of course, when they reach their limits, there are no ways of testing whether or not they're choosing the right answer among the sea of possibilities. Okay, so in case you guys are curious about what is this progression, you easily find it in the internet. If you put 1, 3, 15, 100, here it comes. It doesn't have a name, it has a number in the general files of mathematical sequences. But it's basically 2n minus 5 double factorial, which means double factorial means a productorium of n3 to t, and t is the number of terminals. So if you keep doing this over and over again, you guys know what this means, right? This is summation, but instead of sum, it's a product. Okay, so you start with 3, and then you apply this. You get a number. You get the first number. Then you multiply it by 4, and you do the same thing, iteratively. So you keep getting these numbers. What's the logic of this? For, let's say you have 3 terminals, okay? Like this, A, B, C. Notice that here we actually have 4, because the root counts as 1. For this one, how many possibilities we have of combinatorial possibilities at the tips? A can be the sister group of B plus C, or B can be the sister group of A plus C, or finally C can be the sister group of A plus B. Right? There's no other possibility strictly bifurcating. This is it. They are all rooted. So now think, if we had 4, which means in reality mathematically 5, because the root, as I said, is a 4th one here. How many possibilities would you have of including a 4th terminal here? Well, it could come, let me see if I get another color, so that's clear. It could come here, it could come here, or here, or here, and finally here. So you have 5 possibilities for the inclusion of a 4th terminal D. So you have 5 here, and 5 here, and 5 here, and all of these are different. So you have 5 times 3. So that's where you get the number 15 there. Okay? So it goes on like this. The logic is this. Of course, there are actually more trees than this, because these are only strictly bifurcating trees, and not all trees are bifurcating. You can have trees that have multifurcations, things like this. There's nothing in the method against this. It happens all the time. This is actually a very common phenomenon. Not always we have strictly bifurcating trees. One thing I didn't mention yesterday, but in terms of terminology, it is important. Phylogeneticists, people who work on these things, they refer to fully bifurcated trees as fully resolved. And when you have a non-bifurcating event, like 3 or more, you call it an unresolution, or polytomy. Okay, that's just the way people say things. Why that? Because when you try to disentangle a phylogeny, you try to go for maximum resolution. What's maximum resolution? Is when you can tell the sister group of everybody who is in there. Sometimes you can, sometimes you cannot. Sometimes simply the phylogeny was different, it was not bifurcating. But you try to squeeze as much information from the data as you can. And when you reach full bifurcation, that's when you say that's fully resolved. Because you cannot go beyond that. That's the maximum resolution you can have. Okay? Oh, there he arrived. Too late. Yeah, that's it. Yeah. It's, so this is actually a double factorial, right? It's, if you just take this, that's how they do it. Yeah, that's it. Yeah.
Thank you.
Sure. I was expecting you to give me the answer. Well, it's okay.
I knew it was a function. There you go. That's
Yeah,
as close
yeah.
as I got.
Iterative function. Yeah.
Yeah.
Anyway, so this is just for people to understand a little bit of the mechanics of this. Since for many people this is the first time they're getting in contact with tree thinking, it is interesting to see how we build these things. Now, I mentioned yesterday also that there are 2 main methods for inferring phylogenies, or building trees on the basis of data. Where is the device here? Okay, and I associated them with these 2 people who were the creators of the respective methods, Joe Felsenstein and Steve Ferris. The 2 areas are very different, as I mentioned yesterday. Again, one of them is based on statistics, the other is based on parsimony, but it's important to notice that the justification for parsimony, which is basically minimizing the number of steps, was entirely statistic. It's simply that in the 70s, Steve made all the calculations and reached the conclusion that if you just count the number of steps, you reach the maximum likelihood tree most of the time. So why go through all that trouble? Just choose the shortest tree. You will be right 99.9% of the times. It will just work this way. But eventually people began to take issue with that 0.1% that doesn't work. And actually they dissected exactly what happens that causes this difference. Because the 2 methods are very different in terms of their mechanics. But they reach the same result almost always. So it doesn't really make that much of a difference in practice. I'll show you a couple cases where it does. Let me go for this figure, will come again. This I showed yesterday too. It's just an explanation of parsimony on the basis of bases, DNA bases along a DNA molecule. Okay? For those who are not quite grasping how we do this, let me go through this in a little more detail. Genetic information is coded in the DNA molecule. It can be coding or non-coding. Sometimes it doesn't code anything. Sometimes it does code proteins. So you have these two, and there are more things. There are regulatory areas and so on. So it's rather complicated. But in the end, the data are basically a sequence of letters. These letters are just bases, are just codes you give for specific DNA bases, adenine, guanine, and things like that. Okay, there are 4 of them in DNA. So when you code data for phylogenetic analysis on the basis of DNA sequences, what you have is a string of letters. You have A, C, C, G, T, T, just I'm putting anything that comes to mind here. That's what you have. And this refers to species 1. Let's put it this way. When you have a second species that you want to compare with this one, you also have an equivalent string of bases for the same gene. Okay, it has to be for the same gene, of course, because you have to compare equivalent things. And there's no ends to this thing. I mean, the whole genome is billions and billions of letters, so you choose a few genes that you use to infer phylogeny. So a second species may have the gene, or that part of the gene, the same size or a different size. Usually it is a different size. So you need to do something first, which is called alignment. So you get 2 genes which have different sizes, and you spread one of them so that it fits into the longer one. So for example, in this case, if we had A, C, and then you have nothing here, T, T, A, G, G, C. Okay, these 2 sequences are aligned. What does that mean? That you actually put them in a way that you can make exact comparisons between the sites of the bases. These sites are considered homologous, which means they actually are equivalent evolutionarily. They are a position, okay? Because if you don't do that, what happens is that you end up lining any 2 sequences. Because you have a sequence which is this length, and then you have another sequence which is this length. This one, you just fill the gaps here at the tip. And you end up comparing things which are not exactly comparable. Like you're comparing legs with ears, if you're making morphological phylogeny or something like that. So comparisons have to be between homologous sites, okay? This is a very technical area. There are ways of circumventing it, but I won't go into these details. But in the end, what is informative are the differences, right? Here you have a difference, here you have another difference, the rest is the same. Here, how we count this? What is this? Well, notice, these things are called gaps, or indels. They are not physical phenomena of the DNA molecule. They're just a result of our attempt to align them, okay? So they don't really exist. They exist only on the table. The genes just are continuous, of course. The bases are continuous. So what do you do here? Well, we have to think what's happening. When 2 sequences for related species have different lengths, this can be the result of one of them getting longer or the other getting shorter. That happens all the time because there are insertions and deletions all the time in the DNA molecule. So what we're doing here is detecting one of these phenomena and saying it occurred here. Either the sequence above inserted this C in this G, or this one here below deleted 2 things. So it is a phenomenon, an evolutionary phenomenon as well. Something happened there. And as such, it can carry phylogenetic information, because it can have major portions of the genome deleted, and this lack of this part of the gene is inherited by the descendants. So this can be informative as well, okay? It's something that happened along the history of the sequences. Okay. So it is this kind of information that we use to reconstruct phylogenies. We have to say how this sequence transformed into this one, or vice versa. As I mentioned yesterday, the analysis is done in an undirected manner. So we are not actually looking for trees that look like that. We're looking for trees that look like this, because it's computationally a lot simpler. Not to our minds, because our minds function in terms of sets. We like to think about sets of things, groups. When you're here, you don't have groups. But for the mathematical part of it, this is a lot simpler. Of course, you have many more terms, and things can get very complicated, very big. But you search the best tree on the basis of this. Okay? So once you have all these things done, And these things go on for thousands. It's not just a little bit, it's a lot. You compare site by site. Each one of them is a bit of information that you have to construct the tree. In parsimony, what you do is just choose the shortest tree, like in this figure here. This is the best tree in this case, because it places two A's together and two G's together. Any of the other alternatives will require two independent transformations, either for G or for A. Okay? So it is the nucleus, the logical nucleus of the thing is very simple. It gets complicated as you go along, of course. In maximum likelihood, things are very, very different. What you do is I'm sorry, this example we saw yesterday, I don't need to repeat it. In maximum likelihood, you just estimate probabilities of change between the bases. So if you have a tree, you're not only going to see the number of steps, but you're also going to see the probabilities for in each of the branches of the base in that particular site changing from, for example, A to C. You have a probability here. And this probability is computed in the calculations. And you do that for each branch, each node, each terminal, for each site, for each tree. So, as I mentioned yesterday, this was not computationally possible until the late 90s. For many years, we could only resolve things that involved only 4 taxa, which is nothing. Right? It's just a theoretical possibility. All the rest was just a huge margin of error. But now we're doing better. Now this is actually one of the most used methods today, statistical inference. But of course, it has its limits too, because even today, for very large problems, it gets very difficult to calculate. So people began to use in the past 15 years or so a lot of Bayesian inference based on Markovian chains and things like that to estimate parameters more efficiently. It is very, very used, although the full properties of this are not completely understood yet. We're still making progress on the theoretical part of Bayesian inference.
One question. This probability comes from...
Oh, okay. Where does the probability come from? The probability comes from many things combined together. For example, what's the probability of having an A at a specific site? Oh, not here, because here you know it's C or A or whatever, but in a node. Well, initially the probability is just 1/4. So, because there are 4 bases, right? So you can start with 0, you know. That's the probability of having an A anywhere. But of course, that can be modified, because sometimes you see sequences that have lots of A's, only a few C's, some G's and T's. So these probabilities change. Next, you have probabilities of change, of, for example, A changing to a C. And what is this probability? That's how we see these probabilities here, not always the same. The models get progressively more complicated because they account for different probabilities of things, of change. For example, you know DNA, the bases, they fit each other according to their size inside the molecule. So you have a purine and a pyrimidine together. So changing a purine for another purine is easier than changing a purine for a pyrimidine. Because if you change one here, mutation on one side, if it is from a former purine to a pyrimidine, you need to change the other one as well, otherwise it doesn't fit. It's just too big. So the likelihood of change is also different from one base to the other. So this is taken into account as well. Okay. What else? So you have frequencies of bases, you have transition, this is called transition-transversion ratio, because when you don't change the nature of the base, it's a transition. If you change the nature of the base, it's a transversion. So sometimes you even eliminate completely transitions. For example, you give them probability 0. So it's this kind of thing. In Bayesian, you can put a lot of additional stuff to facilitate the search in this sea of parameters as you make the analysis. Okay? So in the end, the formula for probability is something this big. You just compute all these probabilities. It is quite amazing, actually, that at the end, you usually get the thing simply by counting steps and the same results. It's pretty good. Anyway, these models can get progressively more complicated, but notice one thing, and that's, for me, the major weakness of these models. Once you adopt one model, you use it throughout the tree. So it doesn't matter if you you're treating, sorry if this is getting a bit too technical, you guys can ask questions anytime you want, but I thought this was important to give you an idea of what kind of work is involved in this. Let's say you have a very large tree with many things, eukaryotes, something over 1 billion years old. The probabilities of change of genes here can be very different from the probabilities of change here. So if you use the same model for everything, you're assuming homogeneity of parameters, which is demonstrably not true. That's why very large analyses often come up with strange results, because these models assume uniformity of parameters, and they're not. Evolution is not like that. Rates change all the time, especially over large intervals of time. So you have to adjust these things. Parsimony more or less accounts for that, because you're just counting steps. So the number of steps you count to result here may be very different from the number here, and more or less you allow some flexibility, to account for these changes. Heterogeneity of rates. Okay, so there are ways of accounting for all of this. You can actually compare results using some parameters and using other parameters and see how they resolve or not resolve things, and compare with other data, and you can refine what you're doing and see if it's working or not, where you're going. And there are simulations too. A lot of simulations are done with this. You just put 2 sequences to evolve under several different models and see which method recovers the right phylogeny each time. Okay? All right, so that's how we get these trees. It's using one of these methods. There's no other way. There's one other way I'm going to talk about. This we saw yesterday. This is how the matrix looks. We also saw yesterday. So now let's look a little bit into what's the difference between when the two methods differ. That's interesting. They differ. They give different results when you have one particular situation. When you have branch lengths. What mean branch lengths? So what is these? What the length of this means? This means there's change, amount of change here. So this branch changed a lot, and this branch changed very little. It has nothing necessarily to do with time, because some lineages can change very rapidly and some others can change very slowly. So this one can be very old. It simply didn't change, okay, in terms of mutations. Okay, the problem is when you have long branches and short branches mixed together in the same tree, like in these examples. Why is this a problem? Because as the sequence gets very long, it begins to get repeated hits. So you had something changing from A to C. As time goes by, you have another mutation changing that C to A again. So eventually you begin to have an underestimate of divergence. Because sequences, if you get 2 sequences which are identical, and randomly you begin to give mutations to them and count the number of differences at each T, initially they diverge very quickly. But as time goes by, they don't diverge very much anymore. And eventually they get completely randomized, and time can go by and nothing happens. They stay with the same number of differences. So the equations are not simple. They have to account for this curve of saturation, as it is called. In the beginning, the sequences, you can just count differences and it's perfect. Gradually, you see the time is going by and the differences are not accumulating anymore. So you have to account for this. And when you reach here, you can look as much as you want. You're never going to find any information. It's gone. That's going to mean you're saturated. So you have to choose genes that evolve according to the problem you want to solve. Genes that evolve very slowly, like ribosomal DNA, they are good for inferring phylogenies at very deep portions of the tree. While, for example, mitochondrial, which changes very fast, can be used even for populations of the same species, because they change so fast. Okay? That's why we choose different genes to solve different problems. So today we are being flooded with genomics data, with so much data that is, as I mentioned yesterday, is something we cannot yet handle very well. So we're being a bit clumsy on that. There's too much data, and we don't know what to do with it. Most genomic architecture is not yet known well enough for you to understand what you're using. Okay? Eventually it will. Just one interesting historical bit. When sequence analysis began to be used widely, like in the 80s, people got very enthusiastic, but not knowing any of these problems. So you get these papers proposing the craziest relationships. And people are like, huh? What's happening? Yeah. Basically, phylogeny is not in the molecules because it's so weird. Simply because people are not accounting for all these problems. And so you're getting very funny results. Now that people understand a lot more about the dynamics of molecular evolution, things are getting better. So you say, okay, this is such and such group. You know, they're not putting the chicken along with the earthworm as a sister group of the earthworm and things like that. We are getting more reasonable things that agree more with what we know about evolution from different sources. Okay? So anyway, what happens is this. Why the two methods give different results when you have long branches? Because parsimony just counts steps. So eventually, it will make long branches come together as sister groups. Why? Because you have coincidental mutations here. They're not actually the result of inheritance. They're the result of hits in the same spot that give false similarities here and attracts them together. This is called long branch attraction, precisely for this reason. But one has to consider that sometimes the correct phylogeny can be like this, right? There's nothing wrong. Two long branches can be really sister groups. And then that method will be correct. It will give you the correct answers, always. While the other one will do the opposite. If the phylogeny was not this one, but was rather something like this, then the statistical inference method will give you the right results. But if the actual phylogeny was this, it would artificially separate the two, giving you long branch repulsion. So the two methods have problems. Of course, this is a small percentage of the cases, but that's where people concentrate. Know why they're different. Let's keep going. For example, I mean, for entomologists, this is a very famous example. For people who don't work on entomology, I guess it's kind of weird. But this is very important. I have papers in Science discussing this. The position of this group of animals called Strepsiptera, which you're probably going to live your whole lives and never even get close to one, ever. But they are very interesting. They're very small, very rare, and nobody knows what they're related to. They're insects, they're winged insects, and they may be related to flies, to dipterans, or they may be related to beetles. And the two methods give exactly the two results. Different. Both of them highly corroborated according to the statistical methods we have to testing the robustness of hypotheses. And they are very different, yeah. Here we have for maximum likelihood, you have Strepsiptera related to, these are beetles, okay, so Coleoptera, while flies go elsewhere. If you use parsimony, you have dipterans and Strepsipterans coming close together. Okay, so these are the same data, just different methods. And so in some cases they give you different results, which are quite dramatic because the taxa involved are important. But notice here, the branches are rather long compared to the rest of the tree. So Strepsiptera are long branch, so lots of mutations here. Dipterans, which are these two, are also a long branch. So people always suspect that this might be a long branch attraction. And that the maximum likelihood approach might be better. Actually, the latest analysis of this was last year. It actually put Strepsipteran with Coleopterans. So apparently the maximum likelihood result is right. But of course you have to go, let's say, some reservation about this because genomic analysis is still in its infancy. So there are important cases where the two methods will give different results and people think it's relevant, the difference is relevant. Of course, one very simple way of testing this is simply to do the following. You get the analysis. Okay, you suspect this is long branch attraction. Rerun the analysis without one of them and see if the other one comes at exactly the same spot. And then revert, delete this one, put this one back, and see if it comes at the same spot. If they both come at the same spot, it cannot be long branch attraction because they cannot attract each other in the absence of one of them, right? So the effect would not be there. All right, so there are ways of, ingenious ways of testing for this kind of thing. This I showed yesterday too. It's just the performance of each of the methods for a given set of data under simulation. You see that some methods are really, really bad. You can have a million sequences and they always get the wrong results, while others converge to the right results very quickly. And not surprisingly, these are the methods that we use today, the most used ones. Okay, this I showed yesterday. Now let me just approach something that you guys may find interesting, because it explains a little further what I said yesterday about similarity not always equals relationship. Because this may sound a bit counterintuitive for most people. Like usually things which are evolutionarily close, they also tend to be similar, right? So why not could you adopt some form of similarity measure to infer phylogenetic relationships? It sounds reasonable, but if you think about it a little bit longer and you want to quantify similarity, you see that similarity is actually a function of the characters you see in each terminal. So let's say you have a given phylogeny. Okay, you have a, I'm just making this up, and you have these things here, and then you have an additional taxon which is in the phylogeny, and which will come, let's say, here, and has a very long branch. It means it changed the lines. How we count difference? Each of these tickles, as I said yesterday, represents one change. Say a DNA change, anything you want, it doesn't matter, an evolutionary change. And the tree is kept together by these changes. That's how we discover the tree is the way it is. If you count differences from each of the terminals to each of the others, you make a matrix of differences. It would be A, B, C, D, N, A, B, C, D. You have a certain number of differences. A to A would be 0, A to B would be a certain number, A to C would be another number, discrete numbers, and so on. If you look at this matrix at the end, you see a matrix of similarity. You say which ones are the closest ones, right, in terms of similarities only. Now, if you have a case like this, where one of them diverges markedly from the others, what will happen to these numbers? These numbers will get smaller for the other ones. So eventually, what you get is one long branch as the sister group of all the others. A wrong phylogeny, because this one is the sister group of these two only. Just because when one of them diverges too much, the others get more similar. Because you have to quantify this somehow, right? So similarity is a very, very tricky criterion on which to base phylogenetic inference. Don't get it was used for some years. In the 60s and 70s, people began to quantify similarity as a means of reconstructing phylogeny, but it soon became obvious that this was not a great idea. And people just abandoned this. And here we can see...
Mario, but it's used for pattern test, similarity. No?
Only in a very superficial way, yes. You can just test similarity and see, well, are these things, their sister groups, are they similar or are they not?
No, I meant for test of patternship, the parents test, to identify if one is son of.
Oh, okay, paternity tests.
Paternity test.
Sometimes I wouldn't trust very much because it doesn't work anywhere. Yeah. In some very special conditions it can. I'm just going to show it to you now. Look at this. If the distances are autometric, or if they follow this pattern, what this pattern means that if you have the same amount of evolution occurring in each of the branches. So if you count if the origin is here, you have 5 changes to D. You will also have 5 changes to C. You will have 5 changes to B and 5 changes to A. Okay, so as time goes by, as time is autometric, if you have a fixed number of mutations per unit of time, you end up with autometricity. That's the only case where distances will work. So the genes have to be evolved in a clock-like manner. In reality, this is a condition that is very rarely achieved. It's very unrealistic. Genes don't evolve like that. It can change 5 here, and then it changes 6 here, and then it changes 3 here. It's all messed up. For the same amount of time, we know that most data do not agree with autometricity. They're not autometric.
So
You can adjust for that, but you won't be able to get the values of the tree back the matrix. So it's very inconsistent.
So if that's rarely achieved, how do people use biological genetic clock?
You have to know that in that particular slice of time, that particular part of that particular gene is behaving clock-like, in a clock-like manner. That's the only way. So you have to, that's actually the result of a lot of research. You cannot take it as a general method of reconstruction. It will not work.
It's used to identify lineages within a species. For example, in humans.
Sometimes you can have things that in that little amount of time it's behaving in a clock-like manner. But most genes do not evolve like that for very long. That's why the molecular clock has to be relaxed, has to be adjusted according to the tree, if you want to actually infer time to a tree. Okay, so similarity, just counting similarities, and this is, fits into the general method of distances, doesn't work. Genetic distances cannot be used as a basis for phylogenetic inference. It simply will not work for very basic reasons. I mean, you can deduce them. You don't even need to get to the data. You just think how evolution works and make the counts, and it's not going to work. It simply gives you the wrong answer many, many times. So nobody uses distance data anymore. Use neighbor joining, which is one which is still a little bit used, but it's it works in some cases, but not all. Okay, so that's just so you know that similarity does not mean phylogenetic relationships. Of course, distances can be metric, which is not ultra-metric, but you can still infer the distances between this. You allow for branch differences, branch length differences at the tips, and this adjusts a little more the data to the known ultrametricity of trees. Okay, I will skip that. That's just a method that we use bootstraps, a general method. I'm sure people here have heard about it. To test the degree of support for specific nodes in the tree, okay, that exists. I will not go into any details on that, but different nodes of a tree or different groups can have different levels of support. So you can, some groups are very well supported, some others are not very much. It's not just the whole tree. Okay, this I showed yesterday. I don't have anything else to add to that. This is just how we make the biological, the molecular clock. We take divergence, human kangaroo, human cow, dog cow, blah, 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 blah. And you'll see that the number of substitutions for each of them grows as time goes by. So very similar things that sheep and goat have fewer fewer differences than horse and donkey. And you can use this thing to construct a to discover the equation that will guide the calibration of your molecular clock. Okay? And this is eventually used to infer the degree of difference in relationship to the time of divergence. You know, there's a lot of calculation going there, but you usually estimate the central point of the tree, and then you extrapolate from that for the others. How you initially get the data? You get the data from divergence in combination with the phylogeny, in combination with the oldest fossil known. Okay? All right. Of course, you calibrate that according to many different genes, in many different phylogenies, in many different taxa, so you can have an idea of how well that gene is behaving as time goes by. It's not simple, molecular clocks. Once someone has done it for your group, it's very simple. You just get the program, get the same parameters, and you get the answer. But if you actually have to do the whole work, you better get a grant, because it's going to take a while. Because it involves several different competences. You even need a paleontologist there to tell you the age of the oldest fossil that can be unambiguously identified as that particular animal or plant. Okay? All right. But once you have all this put together, you can do really great things. For example, I showed this thing yesterday, but I didn't explain it very well. This is a group of frogs. I don't know if you can see it from there. There's several little frogs here. This is their phylogeny. And there's the timing of divergence, done very well. This is the error. Okay, there's some error there, but nothing very alarming. And this is an explanation of how these things came to be where they are. They are spread in Asia and part of Europe, but they are related to clays in Africa. So if you actually get this dating that you got from the cladogram after making all these things, you see that these dates agree with what we know about the dates of separation of Gondwana. So these frogs were part of a biota that lived in this region here, and eventually it got kicked out towards Asia, hit Asia, and once they got there, they spread. And that's why we have this relationship. All the dates fit. It's just perfect. You get dates from the molecular clock, you get dates from the continental drift, from other people, have nothing to do with your study, and you see they fit together with the specific splitting events here. Because when this thing left Gondwana and went towards Asia, the frogs became isolated. So they began to diverge. Once they diverge, they become different species. So the events of splitting here are events that can be dated and correlated with specific events in the geology of the region where they occur. So that's really cool. I mean, the history of these things is all there. That actually gave rise to the theory of out of India, because there are so many cases like that people just realized, well, a lot of the fauna of Asia and Europe actually came from Africa, not via north, but via the arc represented by India, drifting across the Indian Ocean. Everything came there, and when they hit, big invasion of things everywhere. So it's interesting, man. There are many other cases now of this showing the same thing. And you can do that for all over the world now. You just can date things, and you can have very robust phylogenies dated and everything. We are reaching a level of understanding of how these things evolved, which is very, very pleasing. This is another example I want to go there. This is from Hawaii, for example. Hawaii is a cool example because Hawaii is on top of a plume, right? As you said in one. David was talking about the plumes and how these plumes actually make volcanoes. So as the crust goes by over a plume, you have islands popping up. Galapagos is also like that. So the crust moves, the plume is fixed, and you have one island, another island, another island, another island, and the older ones begin to erode and disappear under the sea. The oldest Galapagos Islands is like 2,000 meters below the surface of the sea, and very close to the continent. So these things are actually very dynamic. So once they get colonized, what happens is that animals and plants jump from one to the other in a very predictable manner, because usually it's kind of linear. So what would you expect if you made a phylogeny of these things that was correct? You'd expect this. The older islands would be at the bottom of the cladogram, and the younger ones would be at the tips. And this is exactly what you get over and over again. The phylogeny of anything you do, of course, different animals have different fragilities. So not all of them, you can have a species which occupies all of them without differentiation, simply because it can fly from one to the other. So if that happens, of course, you have no differentiation. But when you have the sequence of events here that you infer on the basis of data, not on the basis of the islands, of course, but on the basis of intrinsic data, like sequences and whatever kind of data you're using, will fit the history of the islands. And the cool thing is, when you see the amount of islands which is under the sea, how many things we have lost already that we didn't even see? Many. We just don't know. This is a process that has been going on, colonizing, recolonizing, 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 ad eternum. And sometimes, the first one is quite close to the continent, for example. It could be colonized by the continent very easily. People wonder, how could a Galapagos tortoise reach an island? Oh, okay, they float, but still it's a long distance. If you fly from the continent there, not necessarily these islands were that far from the continent. Many millions of years ago, they could be closer.
Madagascar is the same?
No, Madagascar is different. Madagascar is not volcanic. It's part of Gondwana, yeah. That's why the fauna is so interesting too, because it was isolated for a long time. It was isolated from Africa. It was actually never really part of Africa. It was part of Gondwana. And then when it started spreading, it never really was together with Africa. It's older. You have lemurs there, which are prosimians. Very old. They survived there exactly because it's so isolated for such a long time, so there's not much competition from other things. Madagascar, New Zealand, these places are extraordinary, exactly because they have been isolated for such a long time. As is Chile. Chile is also like that. Chile, even though it is together with South America, it is separated from the rest of South America by very old things like the Andes to the east, the Pacific Ocean to the west, the cold to the south, and the desert to the north. So you have this little rectangle, which is basically an island, and you have things there you don't find anywhere else, usually at the base of the respective cladograms. Very interesting case. In part of Chile, it may have come from a different geological origin. It just hit there.
The north or this?
The north, no, not extreme north, the central part. Yeah, there are terrains that are apparently extraneous to the rest of Chile. So there may have been blocks of continent hitting into Chile along its history. It's quite interesting. I won't talk about that. Oh, okay, I had to include this. Well, he's not here, so I don't think I'm going to talk about this guy. As you see, tree thinking is, but now he's here, now I'm going to talk about him. Tree thinking has reached a very large degree of reception in the biological community in general. People actually talk about trees in ecology books, history, biogeography, everything. Tree thinking is like the guiding force of biology, evolutionary biology today. People finally understood how to get to trees, how to understand trees and use them to understand specific questions. But not everybody. I mean, this is a very famous person, Thomas Cavalier Smith, and he never did understand tree thinking the way other people have. And since he's so famous, because he works on the really deep, ancient portion of the early evolution of life, and he's very productive. I mean, he's a great scientist, don't get me wrong, but his opinions on this subject are really anti-pedagogical for students who read about the subject, because he really opposes tree thinking in general. He tries to formalize it into things which are basically undefensible. The things that have been refuted many years ago. So I won't go into any details, but he prefers this kind of thing. Basically something you can't understand very well. So I will skip to that. It's not necessary, because the only person who asked about him is not here today, so I might as well skip. So you see, the conclusion of this paper just showed evolution should be allowed to classify, rank, and discuss ancestral groups. What are ancestral groups? Ancestral groups do not exist. Why ancestral groups? What you have is a tree, right? You have ancestral species, but groups do not give rise to other groups. Evolution is a populational phenomenon. If you have a very big group here, eukaryotes, millions of species, you had one species as their ancestor, right? The rest is just diversification. You don't say prokaryotes gave rise to eukaryotes. No, that didn't happen. That's the wrong way of thinking, because prokaryotes are not a cohesive biological entity. They're just a result of history, right? So ancestral groups do not exist. Ancestral species exist, but groups, no. It simply doesn't make sense in real terms. You can't say, oh, reptiles gave rise to birds. No, 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 no. One reptile gave rise to birds. It was the ancestor. We don't know which one. But the group as a whole is like saying, oh, mammals will give rise to some other group of animals in the future. No, it doesn't exist. Mammals will not get together and plan what's the next step in our evolution. We, the dog, the cow, the rats, no, let's see what's going to happen. And let's all get together. No, that's not how evolution works. It will be one species of mammal that maybe when all the others die out, who knows? This one will survive and become something else, gradually. Okay, but the others don't care. Huh?
But Mario, you mentioned that in the beginning of the mutations, it has that horizontal changes.
Horizontal transfer, yeah.
Transfers. So different individuals can change in the beginning of a new tree, right?
Yeah. No. With different individuals, what do you mean by different individuals? Yeah, you can have mutations that spread throughout a population, but you cannot have mutations that spread to other taxa in the same way. They have a different dynamics, because they are already separate, right? So if a bit of this comes into this, it is just a bit of this that came into that. It's not the same. That does not make them the same thing. They're still different. It's just that it makes phylogenetic analysis a bit more complicated if you don't know that. It messes up a bit the signal. But no, nothing that approaches what we're discussing in terms of groups, ancestral groups. But anyway, some people insist. All right, he'll die one day. And just like his arguments have been refuted a long time ago. I mean, the 70s. All these papers are classical things on this. And no, I don't need to go into that. There are books that explain the whole thing in detail. So it's a bit embarrassing. You know, undergrads know these things. So the literature is loaded with these things that don't quite fit very well. The progress in the field has been too fast for everybody to absorb it in the same degree, to the same degree. So there are people living today who were raised in very different traditions, and they're eminent in their own right. You know? Yeah. We should not criticize a great scientist like that. Well, he's there, he has his opinions, and I have no alternative other than to explain why I disagree with him. Let's put it this way. Okay, what is this? Nothing? Oh, okay. You guys know this, this drawing? This is from Wegener, the first guy who talked about continental drift. In the 1920s, nobody believed him. Everybody thought he was crazy because there were no forces known to move continents like that. And part of his argument was exactly how Africa and South America kind of fit together in their profiles. And this is the map that showed that. It's a kind of the way you would intuitively put them together. Anyway, Wegener was not resurrected or redeemed until the 70s when plate tectonics became a field of geology. But what I find interesting is that people who worked on diversity, on biodiversity and evolution, they began to see evidence that was so strong that there was something going on between continents in terms of their biotic composition, that some general explanation had to exist. And at the time, geologists just said, well, it's impossible for continents to move. And then they said, well, there must be some other explanation then. Is it possible for continents to go underwater? The geologists said, yeah, that's possible. And then people began a whole tradition of explaining animal distributions on the basis of submerged land masses, just obeying the general paradigm that continents didn't move or didn't change drastically their profiles. So this guy was one of the most interesting. He's a German guy. He actually lived in São Paulo for many years. So that's why we have access to his books, which are very obscure, but they're wonderfully illustrated. This is History of the Atlantic Ocean. This is Archelaus and Archinotis, which are two hypothetical continents that he came to the conclusion should have existed just because of their biological composition. But look at his map. This is a map that he's superimposed on a regular map, showing land connections between different areas. They are not all at the same time, but he showed them all here as they should have existed at some point. And if we see these things, it agrees perfectly with Gondwana, Antarctica together, and Laurasia, plus a few other things which are also interesting. For example, Madagascar and India. They are together longer than any other land masses. So this guy basically discovered continental drift on the basis of the biological composition of the continents. Just that he was not a geologist, so he just followed the paradigm that continents were fixed. I mean, this map is incredible. I mean, continental drift was there. It's just that he had to move the continents together instead of having these land bridges. I find this absolutely appalling in terms of how much he could learn about the world just by looking at what is on the land, living on the land. His main driving force for questioning the current paradigm then was that people say, well, it may be by chance that we have these similarities. But he said, no, it's impossible that it is by chance, because it's just too many things between these two. It cannot be by chance. This has to have a general explanation. So it's someone very ahead of his time. I like this. I'm not going to show much history as I didn't yesterday, but this I couldn't resist. And he gave names to all these things. Even he deduced the existence of a river along southeastern Brazil based on the similarities he found in the freshwater creatures living here. That's pretty good too. Sorry. It was there. And it's funny that he was very much ridiculed in his time. This is a book about biogeography of the time, and it's what it says. And to measure Von Herring, 1900 papers, and other persons, including Eigenmann, have thought that South America was formerly divided by an ocean gap into two whole separate parts, on which separate faunas of freshwater fishes and other animals evolved independently. But this idea was evidently mistaken. The evidence against it, summarized above, is very strong now. No, it wasn't. The evidence against it was very, very weak. He was right all this time. It's a pity that he died before he could be revenged. He wrote in Science. That's how science looked like in 1900. Quite different from today. Anyway, this eventually became incorporated when continental drift became the new paradigm in geology. This became incorporated into a new theory of biogeography called vicariance biogeography, which united phylogeny, geology, and history in general into a general theory of biogeography, that life and Earth evolved together. The two histories are the same, which was very popular in the 80s and early 90s, and today has been highly modified. It's not even called this name, but it was very important historically, and it actually taught us to think straight in terms of biogeography. I don't know why this is white. Why do I have so many blinks? Oh, okay. That's why. Sorry about that. This was there, but I just picked up the slides, and the sound is there. Let me get rid of this. What is interesting about biological distributions is that they are not uniform. This is a fact that caught Darwin's attention, and was one of the strong points in favor of his theory. Is there a way of putting the volume down here? I don't know, this computer is not mine. Oh, okay. I would just...
Function.
Okay, function. It's probably mute now. But the distribution of animals and plants is not uniform. It changes dramatically from continent to continent, from area within continents, and so on. It's not all the same thing. And sometimes you find things that are striking. For example, this family of fishes, these freshwater fishes, they occur in southern South America, southern Africa, southern Australia, and New Zealand. So why you find, it's not the same species, it is actually a group. So you need to know how they're related to reach this conclusion. Why is this distribution relevant? Because it informs us about past events. Not only informs us about past events, but show how the diversity of life is related to the history of the continents. Because the history of the continents is what actually determines the diversity. It's the driving factor. Okay, we don't need that. If you look at the map at a different angle, you see that their distribution is basically southern. Oh, here it's missing here. This is actually not a taxon. This is Notophagus. It's a plant, but shows the same thing. The same pattern of distribution. It's not here, but on the other hand, it is here in Antarctica as fossils. This is a plant. So what's the logic of biogeography today? When you see all these patterns repeating themselves, you try to look for a general explanation. So this general explanation is the history of the lands where they occur. This is one of the pioneering papers on this, this sort of tradition that tried to correlate the history of land with the history of taxa. So of course, he was a biologist, so the history of taxa came first. It was only later that he incorporated the history of the land. And he did it for this group of fishes, people who like aquarium fishes who know them. And he did a very, very detailed study to the level where you didn't have any more geological information to match the biological information, and came up with very interesting results about the evolution of Central America, which has been in part corroborated over the years. So you have cladograms for these things which inform you about the relationships of the areas, how these areas separated as years went by. And it's very, very detailed. So this is the kind of thing you can do with cladograms in terms of biogeography and relate them to the evolution of the taxa. Eventually you can reach even a tree that instead of depicting relationships of taxa, depicts relationships of continents and land masses, like this one, for example. And you can also date them by independent means. Okay, this is just illustrative of the principle. I'll not go into the details of this because this is unnecessary, just for you to see that this is a very interesting area of research, because you can basically integrate biology, geology, and evolution into a single thing. Okay, this is a group of fishes that occurs in South America, Africa, and Southeast Asia. I'll skip that. I'll keep that for tomorrow. Okay, now, how much time I have? You said you would control time.
You have 25 minutes. 3 minutes to time for question.
Okay, so let's now get into a related subject, which is species. What are species in biology? What is it that we call species? And how this concept relates to the evolution of life in general? Well, most of you will be familiar with this particular concept of species, which is a concept developed in the 40s. Species is a group of populations, or interbreeding populations, genetically isolated from other groups by mechanisms of reproductive isolation. Sounds reasonable, right? Everybody, I think, will agree with this definition, more or less. Things that, specimens that belong to the same species will readily reproduce among themselves. Okay. But how does speciation come from? How does it happen? I mentioned in the first class that speciation happens when one primitive population gets split in two or more by some isolating factor. This isolating factor can be anything. Can be a chain of mountains that just rose in the middle of the population, separated into two. Can be a volcano that cleared up a huge area and made it uninhabitable for most of the areas except one little one that stayed there. Can be an ocean that separated two continents because the sea level rose. Anything can serve as an isolating mechanism. Remembering that different organisms have different abilities of dispersal. So an organism that can easily fly over a river will never differentiate by just the existence of this river. But one which is incapable of crossing this river, like a turtle or something like that, will be isolated by the river. So this varies according to taxa, according to the animal or the plant. And also you have ecological conditions on top of all that, further restricting the pathways that they can follow. But in general, species originate by isolation. And it's really important to remember that evolution is a populational phenomenon. Evolution is not something like, oh, we're here in this room and suddenly someone stands up and screams, oh, I speciate, I evolved, I suffered a mutation, I'm a new species. It gets away, it goes away. No, it's not like that. Speciation is something that would happen, for example, if there was a nuclear winter outside and we stayed in this, well, it would be a little difficult to survive very long, but we stayed in here for 1 million years, just among ourselves, and slowly adapting ourselves to local conditions. And when you got out 1 million years later, we'd meet other people who stayed outside and they would be very different. So that's how evolution works. It's always a populational phenomenon. It's never an individual phenomenon. That's why you see, one of the most stupid remarks by creationists. Oh, if man came from the chimp, already wrong, how come we don't see chimps turn into humans today? It doesn't make any sense. I mean, it's not something that happened like this. Evolution doesn't happen at an individual level. It happens at populations. Populations change. Actual organisms within that population never see any change. Never see any change. They're part of a common reproducing community. That's what they are. That's what we are. We are part of a species. We do not see any change. The change is gradual, from generation to generation. That's what makes evolution, right? Okay.
Let
So
me just ask you one
okay.
naive question.
Oh,
If there is an
those are the best.
individual that received somewhere in the past a massive bombardment of cosmic rays, or something like that, that provokes some mutations,
Okay.
this individual will not begin anything, no?
Do not begin anything. No. No.
Okay.
Let's say the theoretical scenario that you bring about. You have what will rain on? Cosmic rays, you said? Yeah.
Yeah, cosmic rays.
Cosmic rays or atomic bombardment or something like that. What will be the result of that? It will increase the mutation rate. That's it. It will increase the mutation rate. And what will be the result of that? You have more variation. Most of these mutations will be really bad. So the result will not be happy for people who will be born after these mutations have affected the germinative cells of their parents. So most will be defects, genetical defects. Over time, probably what will happen is the species will simply be selected for those with the best mechanisms of DNA repair. because mutations at this level, they're usually deleterious. They're not going to be good. So stabilizing selection will just wipe out the ones that are out of the norm, and eventually things will go back to normal. So nothing is going to change in terms of the rate of evolution there. It may happen that one of these mutations will be positive for something. So in this new environment, this new radioactive environment, there may be variants which are more adapted to the new conditions, and they will spread. It is perfectly possible. This is probably what's happening in the region about Chernobyl. When it exploded, everybody thought, oh, nothing's going to survive. Today is the largest preserve of the region. Some of the largest populations of deer, of bears, are there. Just because humans can't get there. Humans are very bad for nature. We destroy everything. Yeah. So even an atomic explosion is better than humans. Yeah. Yeah, we're really nasty. Chernobyl now is like the sanctuary of things. It's absolutely fantastic. Some of the largest populations are there. In the beginning, things were tough for the individual animals. They suffered a lot. But their offsprings were selected, and the ones who survived are the ones which are particularly tough to radioactive conditions. So they look normal now. They're just back to normal. You just can't live there now. So there was, of course, a period of crisis. They eventually was overcome, and today everybody's very happy. The cooling tanks of Chernobyl are full of fish. Huge fish. They're there, just happy. Nobody fishes them, of course, because they're radioactive. Some of the islands that were used as atomic experiment sites, like Bikini and places like that, today they are some of the best preserved coral reefs anywhere in the world. Just because nobody goes there and they recovered.
So it turns out that nowadays the tendency is to select the radioactive fish.
The what?
Being radioactive is a very good.
It's a very good thing, yeah, yeah. Being a radioactive spot for wildlife today is a very advantageous trait. You will survive. So in terms of the biosphere as a whole, I became a proponent of nuclear energy. It's much better than the others. Anything that just keeps us away is good.
To protect the environment. Interesting.
Just bomb everything.
Yeah, that's interesting.
Yeah, I mean, because it will be fatal in the beginning for individual organisms, but in the long term, it's fine. They get along. They resisted very bad things anyway in the past. It just recovers. But they will not recover if we keep destroying everything like one by one, you know? And destroying the environment beyond repair. So it's very interesting. These accidents and bombardments are very interesting experiments. They actually showed us that nature responds in very different ways. Yeah. It's expected, actually. All this was predictable. What people didn't know was that the levels of radiation would be so high that nobody survived. If you get to this level, of course, there's no recovery. Once the species goes extinct, there is no return, right? But the thing is, it's very strong in one point, but it gets gradually less strong as we get away from it, like Chernobyl, right? So at the site itself, there's not much. But today, life is everywhere there. Well, okay. Why did you get into that? Because of speciation. I was talking about speciation, and your question came about the rate of mutation. The rate of mutation will affect may affect the rate of evolution depending on the selective regimen that the species is subject to. Okay, so it can be an interesting factor. But if it's that high, it will probably be just as good as nothing. Because just too many mutations will just make an organism ontogenetically inviable. It won't be able to function. But of course, if this happens over and over again, millions of times, you end up having one, which is good, which is how mutations operate. They don't know what they're doing, but sometimes something interesting comes about. Okay? So the important thing is to understand that speciation, the process of splitting of species, is a result of external factors. Something separated the species in two. And once they're separated, there are mechanisms that will assure that gradually they will get more and more different from each other. This is certain because of two factors. One of them is selection, which you know very well. We've talked about it. We're going to talk more about it tomorrow. But the other one is drift. So even if you don't have selection, drift will make sure that populations diverge once they are separated. Why they do not diverge when they are together? may seem like a silly question, but there is a mechanism for that. The mechanism is reproduction. They're reproducing with each other, so the genes are being shuffled all over the place. So they don't differentiate because they're being mixed together every generation. Of course, distance can be an important factor. The probability of reproduction among individuals is not the same. Each one of us here is much more likely to reproduce with someone from Rio, someone, well, some of them don't live in Rio, but the one many here do, or from Brazil, or from the New World, than reproducing with someone very far away, in China, for example, right? So the probability change. And sometimes the sheer distance between members of a population can be an isolating factor. There are simulations that show that seem to have actual cases in nature. But overall, you need a barrier, a physical barrier. Or, if not a physical barrier, something that acts like a barrier for the flux of genes, for the flow of genes among individuals there. Once you have that, things necessarily diverge. I said selection is one factor. You guys can easily imagine what selection is. You know, if the conditions are different, selection will favor different variants of the genes here and there. So they will get different because they adapt to different conditions. But even if the conditions are the same, they will drift apart because of genetic drift. What is the genetic drift? It's variation in gene frequencies, which are not associated with any selective value. And they happen in all populations. There are genes which are neutral. They're neutral. They just don't make any difference. They have their variants, their alleles. You can have one or the other, and the selective value is the same. Even in this case, they will diverge in different populations in terms of their frequencies. And the funny thing about frequencies, genetic frequencies, is that they oscillate all the time, from generation to generation. It's like this, literally. Sometimes they reach 0, and the other allele will reach 100. This is the only number that does not come back. Once an allele disappears, it's disappeared. So the other one got fixed. So if you have two alleles varying like this as generations go by, one of them reaches 0. Okay, now you only have the other one. Of course, you can have mutation pressure that will keep coming back, but at very, very low rates. That's not usually very relevant. Okay, so things will diverge no matter what. Even if you don't have selective pressure, things will diverge. Populations will diverge by drift. It's a very important force in the divergence of populations. Okay? So you split. Like I said, if we stayed as a separate population in the National Observatory here for 1 million years, we would be a different species compared to what stayed outside of here. You say, oh, the conditions were the same, blah, blah. Doesn't matter, we would be different. Eventually, we would be very different. And once you get too different, you don't breed anymore, because you don't even recognize each other as partners. Okay? Or if you do recognize, the offspring are not viable, physiologically viable. They're just too different to mix. Of course, this is just, as I said, this is just a result of drift. So sometimes you can have things that breed, they belong to different species, and just by chance the offspring will be viable and sometimes even reproduce. It happens, but it's just not the norm. Usually you diverge to the point of no reproduction. And that's when you have a separate species. Okay.
If you had selective pressure, things would occur quicker?
Yeah. If you have selective pressure, things occur quicker.
So if you colonize Mars, right, as
Oh,
opposed
yeah.
to your observatory example, it would occur faster in Mars because it's so extreme compared to...
Oh, yeah. Oh, that's a good example, yeah. Thanks for bringing that up. It's much better than being isolated in the National Observatory. If we actually manage to colonize Mars, as some people are saying, they're trying to do, getting volunteers and everything. Let's say if they only send 6-5%, it's not going to happen. You need at least a few thousand. So, and they actually manage to somehow stay there and survive and be self-sustainable. Eventually, there'll be a different species, unless travel becomes very easy. But if let's say you actually colonize Mars in a self-sustainable manner for an indefinite period of time, people can just live there, and then our civilization on Earth begins to decay, and eventually you lose the ability of going back there again. Because you don't have money, you don't have the expertise, you don't have anything. Yeah, there'll be a different species in no time, because the conditions there are very different. You have cases like, well, everybody has 20 kids. Of these 20 kids, 1 or 2 survive into adulthood. the ones that are really tough to the right conditions there. And then in a few generations, everybody will look very different. Yes, but you need, of course, a critical number. If you have very few, it's simply not going to happen. Everybody's going to die. Yeah. You need a lot of people for for this to happen, especially considering long generation times. So you need conditions which allow such a big animal to survive for such a long time. But you can survive miserably. People can reproduce miserably. Yeah, it won't be a happy life for a long time, but they will be there reproducing. No. And eventually, yeah, some of them will be certainly better adapted to local conditions than others. And it will be different. Yeah, and then 10 million years from now, another technological species comes. And so what is this monkey doing on Mars? I mean, a big mystery. What is this? There's a question there.
I wonder, actually, when talking about these harsh conditions, if we, here on Earth, for human beings, we consider, for instance, I don't know, people here from Brazil, where the temperatures are higher, and people from Norway, where the temperatures are colder, would you say that there is an adaptation that is different for
oh, definitely that.
For
yes.
human beings, are we?
Oh yeah, not now, because we are too recent here as a population. But if you compare, for example, the Swedes with the Africans, African populations, very different. Yeah. Nowadays, it has been proven that being white is adaptive. Because if you live in Sweden and you're not snow white, you die of raquitismo, okay? Rickets. You die of rickets. It's rickets, right? You actually will not function because the amount of sunlight there is so little, and I mean during the winter there is almost nothing in the higher latitudes. That people will actually get rickets, and they will not function well. So you need to be very white. Actually, if you have in primitive times, of course. Nowadays it doesn't make any difference. This is just a legacy of the past. But the difference is big enough for that. And the opposite is also true. It's just that we appeared in a very sunny place, so originally we have a skin that was adapted to that. But if you get a Swede to live in wild conditions in the middle of the Kalahari, he or she will die of skin cancer very fast, perhaps before being able to reproduce. So the two things are adaptive.
But would that be enough to for those two populations in the past, the Africans and the one that became white, for being in a place where there's no sunlight, to be two different subspecies, two different?
Oh yeah, definitely. If that stayed on for too long, it would eventually reach this level. It's just that it was not long enough.